Hello champions, welcome to 360 out of 360 in NEET Biology 2025 within 250 days series. And today, as I promised, I am going to talk about male reproductive system and spermatogenesis, which comes under human reproduction. Now, at this point of time, if you did not understand the concept, or if you were not able to do one or two questions or the 5 questions that I gave you, it's totally fine. I appreciate your efforts. We shall continue doing this. So as of now in this video, I'll give you a detailed explanation of everything that you need to know. So what are we waiting for? Let's begin. I do not want to waste any bit of your time. So we'll be using NCERT as our reference and we'll start. If there is something extra, I'll definitely teach you then there itself. So first point is the male reproductive system is located in the pelvis region. Children, where is the pelvis region? Do you know where is the pelvis region? If I make Pappu over here. So Pappu is my friend. I make Pappu's diagram over here. I'm not making Pappu's head. So this is the area where your heart is residing. What do you call? You call this cavity as thoracic cavity. Fine. You call this cavity as what? Thoracic cavity. Now below this, what do we have? We have something that is known as abdominal cavity. Abdominal cavity can be subdivided again. So this lower region of abdominal cavity, I can specifically give a name. What is its name? I can call that this is the pelvic cavity or the pelvic region. Now, why is ma'am saying all this thing? Because children, it is written that male reproductive system is located where? In the pelvis region. Is it important? Very much important. So, it is located where? In the pelvis region, in the pelvic cavity, or you can say abdominopelvic cavity. Just combine both of them and write abdominopelvic cavity. Now, what does it include? It includes a pair of testes. Okay, it includes a pair of testes, accessory ducts, glands and external genitalia. Now, children, testes. Basically, when I say pair of testes, this is plural. Okay. If I use this particular spelling, testis, this is singular. I call testis as primary sex organ. Why do I say that testis is the primary sex organ? So what about others which are mentioned over here? Accessory ducts, glands and external genitalia, they are all secondary sex organs. So why do I say that this is the primary sex organ? Because it functions, it has the primary function. What is the primary function? Obviously, the primary function of the male reproductive system should be the formation of sperm. Without sperm, is there any use of this particular system? Obviously no. So sperm, and the second thing is what? It is also responsible for the formation of male sex hormones, and together all the male sex hormones, what can you call it? You call them as androgens. What do you call androgens? So your testosterone, everything is a type of androgen. So male sex hormones are known as androgens. So testis is known as primary sex organ. Why? Because it helps in the formation of sperm and also it helps in the formation of androgens. Clear? Now, if we talk about accessory ducts, glands and external genitalia, we'll be seeing it one by one. Okay, chumma I'll say a little bit function of it. Accessory ducts are responsible for transportation. So someone is making sperm. Who is going to transport it? Accessory ducts. What about glands? Glands are going to give secretions to the sperm so that the sperm become mature, it has proper motility, it can move easily and everything. Okay. External genitalia, that is the external genital organ. It is responsible for insemination, that is for the transfer of sperm from the male body to the female body. You need an external genital organ, which in males it is what? Penis. Penis. Okay. So basically external genital organ in males is what? Penis. There is only one thing to remember. So basically that's why I wrote it. Accessory ducts and glands are a bit long. We will study about it one by one. Okay. So let's begin the battle again. The testes, if we talk about the testes, let's see what all we have to study. They are situated outside the abdominal cavity within a pouch. I said right, abdominal cavity. So abdominal cavity, as in what are they telling? Abdominopelvic cavity. Okay, they mean abdominopelvic cavity because we know abdomen also has two regions. The lower region is pelvis region, below it is present. So I can clearly say it is present outside abdominopelvic cavity. Now it is within a pouch called as scrotum. So if you see this particular diagram, or probably if I make one, if suppose that this is a testis. So your testis is basically present in a pouch. And what do you call this pouch? You call this pouch as what? Scrotum. Okay, called this as scrotum. Now, what is the use of this? This is a very important thing. Scrotum helps in maintaining the low temperature of the testis, that is 2 to 2.5 degrees Celsius lower than the normal internal body temperature. I am meaning to say that if the testis was inside the human body, okay, the temperature is too much, and in this particular temperature it cannot do its primary function of formation of sperm or the formation and synthesis of what? The androgens or male sex hormones. So what is one condition that the testis has asked for? That please put me outside the human body inside a pouch, and what is this pouch known as? Scrotum. And what is the temperature? What is the temperature difference? 2 to 2.5 degrees Celsius lower than the human body temperature. Please remember these small details. Now, in adults, each testis is oval in shape. Okay, the shape of the testis is what? Oval. Now, if you talk about the length and the width, it's very easy to remember. I'll tell you how. So, the width is 2 to 3 centimeter, okay, and the length is 4 to 5 centimeter. Okay, width is 2 to 3 centimeter, length is 4 to 5 centimeter. So you can remember 2, 3, 4, 5. You just skip the one there. Yeah. Now we go ahead. The testis is covered by a dense covering. So testis obviously has lots and lots of covering in there. It's not a free thing. So you have tunica albuginea to cover the testis fully. We go ahead. Each testis has about 250 compartments known as testicular lobules. How many compartments do they have? 250. What do you mean by compartment? Let's see it. So imagine that this is a testis. If I cut the testis in half and I see the sagittal view, the side view of it, I can find 250 compartments like this. How many of them? 250 of them. What do I call these 250 compartments that is seen over here? You call it testicular lobules. What do you call it? Testicular lobules. How can you remember it? TL, team lead. Okay, this is the person that is going to lead the testis. So over here, testicular lobules are there. How many of them are there? 250 of them are there. As of now, you just need to remember this. Okay. So we are done with testis, which is there in the pouch known as scrotum. Why it is there in the pouch? Because we need 2 to 2.5 degrees Celsius lower than the human body temperature. What is the width? 2 to 3 centimeter. What is the length? 4 to 5 centimeter. It has a dense covering. You call it tunica albuginea, and the outermost covering. There are so many muscular layers as well. Now we go ahead. What do we have? Inside the testis, you have 250 compartments. What do you call these compartments as? Testicular lobules. That is TL, testicular lobules. Now let's see what's in there. Okay, so before getting in that, I have a question for you. Okay, let's solve this question and then go ahead. Over here, the testes in humans are situated outside the abdominal cavity inside a pouch called scrotum. The purpose served is for, what is the purpose of this particular thing? Please see the options and choose the correct one. Let's read it one by one. First one, maintaining the scrotal temperature lower than the internal body temperature. According to me, it's right, because obviously we need lower temperature. How much lower? 2 to 2.5 degrees Celsius lower. Now, second, escaping any possible compression by visceral organs as in internal organs. I don't think so that is a possible reason. Providing more space for the growth of epididymis. We have not even come across epididymis as of now. So, let's just exclude it as of now. Providing a secondary sexual feature for exhibiting the male sex. I don't think so it is needed to give a secondary sexual character. So, over here, even if we are not sure about any of this thing, I am sure about one particular option. So, can you go with this one particular option because we have already done it? Yes. So, this is how we study. Okay. Now, this came in NEET 2011. Again, you got 1 marks. Now, if you go ahead, we were stuck at what? What was that thing? 250 testicular lobules, right? Okay. Each lobule contains 1 to 3 highly coiled seminiferous tubules. So, for this, I have to make a small diagram. Okay. So, let's make a small diagram. As I said, how many compartments do you have, children? 250 compartments are there. What do you call them? Testicular lobules. Now, inside that, what do you have? You have 1 to 3 highly coiled seminiferous tubules. So, let's make 3 of what? something that is known as seminiferous tubules. Can I write this? Seminiferous tubules is going to be very easy, something that looks noodleish over inside. Okay, now each seminiferous tubule is lined on its inside by two type of cell. Now they are asking you to zoom in one seminiferous tubule. So let's zoom in one seminiferous tubule. So this is a red thing. I'm just going to make it like this. So this is one seminiferous tubule, and this is going to be the next seminiferous tubule, and they are saying that each seminiferous tubule is lined on its inside by two types of cells called male germ cells, spermatogonia, and Sertoli cells. So are you ready for the battle? Let's be ready. Over here there are two cells. One cell is the male germ cell. What do you mean by germ cell? Male gametic cell, which is going to make the gametes, that is the sperm. So the papaji of sperm is over here, that is the male germ cell. It has a specific name as well. What is the specific name of this? I'll write it over here. I'll make it over here and I'll write it. It is known as spermatogonia. What do you call it? Spermatogonia. Fine. Now we go ahead. What do we have? This is just one type of cell. We have two type of cell. The second one is what? Sertoli cell. So let's make Sertoli cell. There is another type of cell over here. What do you call it? Sertoli cell. Now I'll tell you, there is one more name of Sertoli cell. What is it? What is that name? Nurse cell. And what do they do? The profession nurses, what do they do? They take care of the patients really well. Similarly over here, they are going to take care of the spermatogonia really well, such that the spermatogonia can divide itself, can undergo lot of divisions, and finally give rise to a sperm at the end. So this is a male germ cell. It still has to form a gamete. Okay, this is just the first cell. It will undergo multiple division to finally form a sperm. Now who will provide nourishment to this particular spermatogonia? It will be the Sertoli cells, which with love I call nurse cells, because they are going to take care of them. Fine. Now these are the two cells that is present inside the seminiferous tubules. Let's go ahead. The male germ cell undergo meiotic divisions, finally leading to sperm formation. So meiosis is when you need to lower down the number of chromosomes. Okay, so over here, if I say there are two types of division, one is mitosis, and the second one is meiosis. Okay, in mitosis what happens? Just simple thing I am saying. Okay, I am not making it too complicated as of now. Imagine there is a cell with 46 number of chromosomes. There will be two cells which after division will be still having 46 number of chromosome. Since the number of chromosomes are equal in the parental as well as daughter cells, what do you call this type of division as? You call this type of division as equational division, because it is equal. Equational division. Okay, I just have to rub this. We will write it properly. Equational division. Yeah. And now on the other hand, for meiosis, what will happen? If I have a cell with 46 number of chromosomes, there will be two sets of division that is going to happen. In the first division, that is in the meiosis 1, you will get two cells and it will be having 23 number of chromosomes. Have you seen that the number of chromosomes have reduced? Which is why you call it as what? Reductional division. It is going to reduce. Now over here, if I divide it again, you have to divide it, because meiosis has two stages. This is the first stage where the reduction actually takes place, and this is the second stage, which is exactly like this, where the number of chromosomes will be the same. So 23 here, so 23 will be here as also. Similarly here also it will be 23 and 23. So the question is, why do gametes undergo meiosis and not mitosis? So you only imagine that there is a sperm and there is an egg. So there is a sperm, that is the male gamete, and there is an egg. If both of them were carrying 46 number of chromosomes, together they will give out 92 number of chromosomes. Is this the actual number of chromosomes that we have in our human cells? No, we have 46 number of chromosomes. So basically what do we need to do? We need to make it half. We need to make it half. Let it be 23. Let it be 23, together it will be 46, and tada, it's just like a normal human cell. So over here, if meiosis is happening, the number of chromosomes will be reduced, and one such cell, when it goes and fuses with another such cell, obviously it will be giving 46 number of chromosome, which is why we prefer meiosis. Okay. Now this spermatogonia that you were studying, right, it will also undergo meiosis. So whenever gamete formation is there, the only cell division that takes place is meiosis. You have to remember that. Okay. Now we go forward. While, okay, so the male germ cells undergo meiotic division, finally leading to sperm formation. While Sertoli cells, what do? They provide nutrition to the germ cell. What is nutrition? Okay, I said, what do you call these cells as? Nurse cell as well. Please remember it. Now, the regions outside the seminiferous tubules, that is, if this is 1 and this is 2, the region in between the seminiferous tubules, okay, is called interstitial space. What do you call it? Interstitial, as in the space in between the cells. So interstitial space. Now what is the use of this space? Basically in this particular space there will be lot of cells, and what do you call these cells as? You call these cells as, I'll write in purple color, wait, you call them as Leydig cells. What do you call them? Leydig cells, or you can also call it interstitial cells, since it is present in the interstitial space. Okay, so Leydig cells or interstitial cells. Ma'am, what is the use of it? What do they do? Basically children, they are responsible for the production of the male sex hormones, that is androgens. So we studied that testis is responsible for formation of sperm as well as the male sex hormones. Who is responsible for formation of sperm? Spermatogonia, okay, which will undergo meiotic division. Who is responsible for formation of male sex hormones? It will be the Leydig cells. I hope it is pakka clear to you guys. So let's read it. Okay, so the region outside the seminiferous tubules called interstitial space contains small blood vessels and interstitial cells called or Leydig cells. Leydig cells synthesize and secrete testicular hormones called androgens. Other immunologically competent cells are also present, as in all the white blood cells, different type of white blood cells which could fight against diseases, they are also present over here. And also you have what? Blood vessels. I hope this is clear to you. Now let's go ahead. What do we have? The Leydig cells, as found in the human body, are the secretory source of. That means what are they responsible for secreting? That is a question. Now you have options as progesterone, you do not know it. Intestinal mucus, the mucus present inside the intestine. Okay, glucagon, no, that is a hormone. Okay, if you go ahead, androgens, you already know that it is what? Androgens, that is the male sex hormone. In which year did this question come? 2012. Is it important? Yes. And do you know this? Obviously yes. It's very easy, right? So 2 marks. Now we go ahead. What do we have? It is about the accessory ducts. And what are accessory ducts responsible for? I already told you it is responsible for what? Transport. It is responsible for transport. Now over here I have a picture that is already being made. Let's see it one by one. So male sex accessory ducts include radiotestis, vasa efferentia, epididymis and vas deferens. Is it too much to remember? It's very easy. Trust me. Okay, so let's do it one by one. If we zoom this in, if I can zoom this in, it will be really good. Let me check it. Okay. Okay. Try muddy. Okay, it's not happening. Fine, then I'll do one thing. If it is not happening, then we have a solution for it. Okay. So your testis is there. Fine. Inside the testis, what do we have, children? TL. What is the full form of TL? Testicular lobules. Fine. Inside the testicular lobules, we have noodle shaped what? Seminiferous tubules. How many? 1, 2, 3. Now let's do it. The The seminiferous tubules of the testis open into the vasa efferentia through rete testis. That means there is something that is known as rete testis in between, which will be opening into vasa efferentia. So NCERT always complicates things. Let's make it simple. Okay, so I'll tell you, between that it's not enough, you need to study one more thing, very simple thing. Okay, so over here there is something that is going to carry things out from your testicular lobules as well. What do you call this? You call this as tubuli recti. This is one extra thing, can you remember it? What do you call it? Tubuli recti. Now all the tubuli recti, they will come together and they will be opening into something like this, which you call as, is it visible or should I write in white? I'll write in white. It is known as rete testis. Now as written over here, the seminiferous tubules of the testis open into vasa efferentia through rete testis. So this is rete testis. What is this going to open into? It is going to open into vasa efferentia. So these things that take things away from the testis, outside the testis, okay, it is making things exit the testis. It is known as vasa efferentia. Vasa efferentia, or with love I call it efferent ductules. So please don't get confused, it's the same thing. So what do we have? Efferent ductules. Fine, this is also done. Now how can you remember this? E for exit. So this thing, particular vasa efferentia, there are almost 20 of them. Okay, there are almost 20 of them, and they are going to take the sperm which is going to be made here outside the testis. Now let's see, where is it going to? This particular thing is going to form something else. So the vasa efferentia leave the testis and open into epididymis located along the posterior surface of each testis. So basically this vasa efferentia is going to open into what? Epididymis, which is located in the posterior. This is the posterior side of the testis, and here the epididymis is formed. You might be thinking, why I am making things like this? Because it is super coiled structure. This epididymis that you are studying about, it is a super coiled structure. What is it? Epididymis. Almost it is 6 meters long. That is why it has to be coiled, and it is present around the posterior surface of each testis, not ovary, each testis. Fine. Now, why is epididymis so important? Because epididymis is the place where the sperms basically, they mature and they attain the motility. Okay, they mature and attains motility. Mature as in, aren't they mature already? Because you know what, sperms are not always in the flagellar shape. Okay, they are normal cells, so they have to reduce the amount of cytoplasm to be in that shape so that they can move easily. Okay, so all this thing is partially done in epididymis, and some of the things are also done in the testis itself. So it is responsible for maturation as well as to attain motility, so that the sperm can easily move around. Okay. Now the next thing is that the epididymis leads to vas deferens that ascends to the abdomen. It sounds too confusing, let me make it down easy for you. So we were just talking about testis. I'll just make a smaller figure of it, and over here we have the epididymis, and the epididymis, as said there, it is going to open to what? Vas deferens. So there will be a tube like this which is going towards the abdomen, as said over here, and this said that this particular tube is going to loop over the urinary bladder. So for that we have to make the urinary bladder first. Let's make it. So this is, okay, this is not visible. So this is the urinary bladder. Fine. And the urinary bladder obviously is attached to the kidneys with the help of what? Ureter. Okay, that holds the urinary bladder to the kidneys. So over here the vas deferens is going to loop around this and come front. Okay, so from back it will be looping around and come forward. And it's not all, I'll just write it this first. It is what? Vas deferens. Now the vas deferens, what happens then to it? there will be a gland waiting for the sperm to come, so that this particular gland can give out the secretion. What is this particular gland known as? It has a name. I'll tell you the name as well. So the gland looks like this. Its name is seminal vesicle. What is its name? Its name is seminal vesicle. So this gland opens in over here, and both of them together has a common ejaculatory duct. What do you call? You have ejaculatory duct. Ejaculatory duct. Now, it receives a duct from the seminal vesicle and opens into urethra as ejaculatory duct. So basically, it opens into urethra as ejaculatory duct, which means you know that this is urinary bladder. What is this particular thing at the end? This is your urethra. So basically, the vas deferens and the seminal vesicle together, what are they forming? The ejaculatory duct. And now where is the ejaculatory duct opening into? It is opening into urethra. Now if you take this down, this urethra is kind of long, and at the end of it you have an opening through which the urine as well as the semen goes out, or the sperm goes out. You call this particular opening as urethral meatus. What do you call this as? Urethral meatus. Fine. Now this is basically covered by your external genital organ, which is what? Which is the penis. I hope that is clear. Okay, we'll read it. The epididymis, we were stuck here. The epididymis leads to vas deferens that ascends to the abdomen. It went to the abdomen and loops over the urinary bladder. It looped over the urinary bladder. It receives a duct from the seminal vesicle. This is a gland, accessory gland. Okay, so it receives a duct from the seminal vesicle and opens into urethra as ejaculatory duct. It will be opening into urethra as what? Ejaculatory duct. These ducts store and transport the sperms from the testis to the outside through the urethra. So basically the ejaculatory duct will be storing the sperm and later will be transporting it outside through the urethra. The urethra originates from the urinary bladder and extends through the penis to its external opening. What do you call this? Urethral meatus. That is the end point. Okay, through which the urine as well as the sperm will be moving out. So are you able to understand that in males they have a common opening? Okay, that is the reproductive system as well as the excretory system. Both of them has a common opening, whereas in females it's not the case. And what is this common opening known as? It is known as urethra. I hope it is crystal clear. So if you are going one by one, what do we have? We have tubuli recti, that is the first thing. Second thing will be rete testis. After rete testis, it will be exiting outside with the help of vasa efferentia, E for exit, and E for efferent ductules. Okay, now it will be coiling around. It is known as epididymis on the posterior side. Yeah, and the epididymis will later lead to vas deferens, which will be going up around the urinary bladder. It will receive a secretion from the seminal vesicle. Together it will form ejaculatory duct, and then it will open into urethra. The end of urethra is the urethral meatus. I hope this is clear to you. Now the accessory ducts are also clear. We'll go ahead with the next thing. And this is the picture. Okay, let's escape. Let's leave it for now. Over here you have a question. Which of the following depicts the correct pathway of transport of sperm? First one, rete testis. Okay, we can include wherever rete testis is there, we can see that might be a correct option. So rete testis is there. After that, what do we need? We need vasa efferentia or efferent ductules. Let's see if it's there. So second option is efferent ductules. Oh, I see, it's correct. What about epididymis? No, it's not, because you have so many things in between. No. Vas deferens, obviously no. This is obviously wrong because rete testis should be first. So what can I do? I can cancel out options. This option is cancelled, this option is cancelled, this option is not cancelled. This would be correct because rete testis, after that efferent ductules or vasa efferentia, and then epididymis, and then vas deferens. How can you remember it? You can at least try to remember this particular code, that is RVEV. Rete testis, vasa efferentia, epididymis, and vas deferens. The vasa efferentia and vas deferens might sound same, but D for distance. You can remember like that. So it is at the last. Yeah. So that question came in NEET 2016 again. We go ahead, we talk about the accessory glands. For that, I'll definitely need a diagram. So let's make it. Over here, over here, yeah. The male acidic glands include paired seminal vesicle, a prostate and a paired bulbourethral gland. So over here, as I said, you already know seminal vesicle is there, yeah. Now seminal vesicle and the vas deferens will be opening into what? Ejaculatory duct, and later it will be going to the urethra, right? So around this particular thing you have something like this which you call as prostate gland. What do you call it? Prostate gland. It is not a paired thing. You can just remember, think of a donut, in between that there's a gap, right? So through this gap the urethra and everything goes up, okay? So basically prostate gland is just around it, yeah. And what is the use of it? I'll tell you. If you later go down, you will be seeing small bulb shaped thing which you call as bulbourethral gland. What do you call it? Bulbourethral gland. It is near the urethra and it is bulb shaped. So bulbourethral gland, with love you can also call it what? Cowper's gland. Cowper's gland. Fine. Cowper's gland. That is also possible. Now what is the use of all of them? So seminal vesicle, I'll just write it. Seminal vesicle, it is responsible for giving the sperm fructose. What do they give? Fructose. And what is the use of fructose? It provides immense amount of energy for the sperm to move. Okay, it is a mode of nutrition for the sperm. Okay, so fructose is a mode of nutrition, and who gives it? Seminal vesicle. If we talk about prostate gland, it is responsible for the formation of calcium. Yeah. And why do the sperm need calcium? So sperm has to basically move towards the ovum. For that it needs calcium, and also for the motility it needs calcium. Okay, and there are so many functions lingering around calcium. Now calcium is needed. Apart from that, the prostate glands are also responsible for secreting certain enzymes. What do you call these enzymes? As clotting enzymes. So since the seminal vesicle gave the sperm fructose, the sperm is like very active. But is there a need to be active inside the male reproductive body? No, because it has to go to the ovum in the female reproductive body where it needs motility. So as of now it has to stay at one place. So for that clotting enzymes are released so that the sperm will not lose the energy and will stay at one place. Okay, that is the use of prostate gland. Now if we talk about bulbourethral gland, it is responsible for lubrication. Okay, it is responsible for lubrication, so that it allows easy movement of sperm. Okay, that is all. Now let's read it also one by one. Secretion of these glands constitute the seminal plasma. Yeah, so together the secretion, all of them together, what do I call this? I call the secretion as a very important thing. Okay, I call the secretion as seminal plasma. What do I call it? Seminal plasma. Fine. Now, okay, which is rich in fructose, calcium and certain enzymes. Will you remember fructose, calcium and certain enzymes? It is very important. The secretion of bulbourethral glands also helps in the lubrication of the penis so that the sperm can easily move. Okay, I hope this is clear to you. So we have three types of acidic ducts, acidic glands as well, and one is unpaired, rest all of them, both of them are paired. Will you remember that? So what is that unpaired thing? It is the prostate gland, single. Okay, and the bulbourethral gland is also known as Cowper's gland. Let's see, we might have a question for the same. No. Okay, now there is an important thing over here. Just give me a second, let me erase this. Okay, sperms released from the seminiferous tubules are transported by the accessory ducts. You know that. What are the accessory ducts? RVEV. Okay, secretions of epididymis, vas deferens, seminal vesicle and prostate are essential for maturation and motility of sperm. If you are not giving all this thing, obviously the sperm will not be able to move as well as mature. The seminal plasma along with sperm constitute the semen. So basically the secretion that is known as seminal plasma, if I add sperm to it, this whole thing becomes what? Semen. So the semen which you talk about, it is made up of sperm plus the seminal plasma. And what is the pH of this thing? The pH of the semen is very important. The pH of semen is almost 7.3 to 7.5. Okay, it is alkaline. It is alkaline. So in acidic pH it cannot survive, that is the sperm. So basically when it enters the female body will be acidic, okay, so that it can kill any germs that could enter the genital organ. So basically, to protect the sperm from all this acidic nature, you have a covering of secretions basically which will be alkaline. So together the pH is 7.3 to 7.5. It is an alkaline pH. It is very important for you to remember it. Yeah, so we are done with that. I hope you're clear with that. We go ahead. Question. Given below is a diagrammatic sketch of a portion of human male reproductive system. Select the correct set of the names of the parts labeled A, B, C, D. So you tell me the names of A, B, C, D. Starting with A. It's something that is coiling around the urinary bladder. What is that something that coils around the urinary bladder? I suppose it was vas deferens. So yes, it will be vas deferens. So let's cancel out the options first. So it is A is ureter, A is ureter. Let's cancel it out. Ureter is something that comes here, right? This is ureter, which is bringing the urine from the kidneys to the urinary bladder, right? So this is A, and A obviously will be vas deferens. Now let's see B. B, you know that the first gland, if you remember, SPB, seminal vesicle, prostate and bulbourethral, that is the positioning of it. So B over here will be obviously seminal vesicle. C is something that is making a round over there, it will be prostate, and bulbourethral looks like bulb, and this is a bulb shaped thing. So this will be the correct option. I hope it is clear to you. Okay, is it this correct option? Let's see. B is seminal vesicle, C is what? Prostate, and D is what? Bulbourethral gland. Okay, sometimes no, they will confuse you and they'll write D over here and C over here. Please don't go with that flow. Please try to understand what is D mentioned as, okay, and then only try to answer. So you're done with 2009 question as well. NEET question, so good. I hope you are also feeling the josh of it. Now we will talk about the male external genital organ. What is the male external genital organ? It is the penis. Now over here it is made up of special tissue that helps in erection of the penis to facilitate insemination. So obviously if it does not have erectile tissues, do you think that the sperm can inseminate? No. So the enlarged end of the penis is known as glans penis and is covered by loose folder skin known as foreskin. So this thing I think I have to make it and show it to you. So basically just imagine, if this is the penis, it has an enlarged portion which you call as, what do you call this portion as? Glans penis. Now always remember it has a covering, okay, it has a covering. What do you call this covering as, children? This is totally attached to the penis. This covering is until this particular position. What do you call it? You call it as shaft. Okay, now from here the covering is detached, it is not attached to the glans penis. From here it stops, just the last point of attachment is this. Here there is an attachment like this, double folding, okay, which is not attached in this particular surface. It is just last attachment is this. You have to remember that. Now this particular thing that you see over here, what do you call this? There is a loose folder skin that surrounds the glans penis, saves it, okay. It is foreskin. It is foreskin, and foreskin is richly supplied with nerves which helps in sexual erection. Now that is done. If you see inside the penis, you have 3 sets of erectile tissues. What do you have? 3 sets of erectile tissues. And you know, in between, what do we have? You have the urethra, and at the end you have the urethral meatus, right? Now there is an erectile tissue through which this particular urethra passes. So if I cut this like this and see, for example, if this is the penis, I cut it and see this particular view, okay, I'll be able to see something like this. Where shall I make? Okay, so this particular thing, this particular erectile tissue is through where the urethra passes, okay, and what do you call it? You call this particular erectile tissue as corpus spongiosum, okay, corpus spongiosum. Fine. Now these both erectile tissues, what do you call it? Corpora cavernosa. Don't think, oh ma'am, something extra we cannot study. Don't think like that. Everything is important. Okay, these are known as corpora cavernosa. These are all erectile tissues. Why are erectile tissues very important? Because these erectile tissues are richly supplied with blood vessels inside, blood sinuses. Now, what happens is that basically when the blood enters the arteries, that is when these erectile tissues get stiff, and that is when the particular what's the external genital organ, that is penis, it becomes erect. So it is very important that erectile tissues are there because it does have blood filled sinuses. Now in this, when the blood fills up, it becomes erect. I hope it is very clear to you. So what do you need to remember? The enlarged portion is glans penis, then covering it we have the foreskin, which has the last point attachment here. Okay, and this skin that is present over here is known as shaft. It's totally attached. Now inside this you have 3 erectile tissues. What are those? 2 of them are known as corpora cavernosa, and 1 of them is known as corpus spongiosum. Through corpus spongiosum the urethra it passes through, and also you have blood sinuses inside, through which arteries, when the blood fills up, the erectile tissues obviously they will be active, and over here the penis will be erect. I hope it is clear to you, and with this we are done with the male reproductive system. So let's see. Okay, there's no question over here. Let's see. Okay, fine. We will be seeing question towards the end then. Okay. Now we go ahead. We are going to talk about spermatogenesis. Again, very important topic. So why am I speeding up things? Because we have to utilize time efficiently. As soon as we can complete the 4 pages, we are trying to do that. Yeah. So let's read it one by one, and then we will refer to this particular diagram. The primary sex organs, that is testes in males and the ovaries in the females, produce gametes. You already know that. Okay. Now, by the process that is known as gametogenesis. What do you mean by gametogenesis? You divide the term gameto and genesis. Gametogenesis. Now, genesis means the formation of something, the initial step of something, and gameto, as the name suggests, it means gamete, the germ cells which are going to fuse together and form the zygote. Okay, so the formation of gamete is known as gametogenesis. Now if I replace this term with sperm, I say, instead of gameto I say spermato. Spermatogenesis means what? The formation of sperm, nothing else. Okay, it's very easy. Now they say that in testes, the immature male germ cells, that is spermatogonia, which we studied previously, okay, spermatogonia produce sperm by spermatogenesis that begins at puberty. So at puberty the bell rings, yeah, at puberty the hormone synthesis starts off, which is going to tell that, okay, spermatogonia, now you have to divide yourself and form these sperms. Now this whole process of spermatogenesis we are going to see it right now. So the spermatogonia, singular one, the single one is known as spermatogonium. It is present on the inside wall of seminiferous tubules. We learned it, right? And along with it, who is there to supply nutrition? It is Sertoli cells. Okay. Now, they multiply by mitotic division and increase in numbers. Now, you only tell me, in mitotic division, do they decrease the number of chromosomes? No. It is just for increasing the number of cells. So, whenever I have a wound in my body, the cells, they have to be filled with cells. No. So, here, mitosis will be happening. Meiosis happens only in the gametes. So, this is also gamete mother cell, ma'am. I know that, but it will come, meiosis will be coming. As of now, we have to increase the number of cells which could later give rise to sperm. So, over here the first step is the spermatogonia that you see over here. It is going to increase its number by the process of mitosis. That is the first step. So, this step, it will be known as mitosis. Now, once mitosis is done of spermatogonia, what do you get? You get 2 types of cell, A and B. What do you get? A and B type of cell. Now, each of these cells has 46 number of chromosomes. Okay, each of these cells has 46 number of chromosomes since it was mitosis only. So, I say that it is diploid. What do you mean by diploid? It is suggesting the ploidy of the cell. That means it is 2n. What is n over here? N is 23. So 2n means 2 into 23, that is 46. So all of them, they have 46 number of cells, 46 number of chromosomes, right? So this is diploid, this is diploid, this is diploid. Fine, because they underwent mitosis. Okay. Now let's see one by one. Okay, spermatogonia present on the inside ball of seminiferous tubules multiply by mitotic division and increase in number. It was just to increase the number. Fine. Each spermatogonium is diploid, okay, and contains 46 chromosome. Okay, so this is 46, it has 46 number of chromosome and I am okay with that. Let me write. It has 46 number of chromosome and it's okay because it is diploid. Now, some of the spermatogonia called primary spermatocyte periodically undergo meiosis. So, where did this come from? Some of the spermatogonia, what do you mean by that? Basically, what I mean by that is, once the spermatogonia undergoes division, I said A and B type of cells are formed. This is A and this is B. Okay. Now, what happens? The B1 is selected and it undergoes certain differentiation. Differentiation means it attains a particular function. That is the meaning of differentiation, not cell division or nothing like that. For example, if I decide that I have to become this, I am getting differentiated. You can think like that. Okay. So, basically this particular B cell is undergoing differentiation and now it has certain changes in itself such that it is going to form a type of cell which is known as primary spermatocyte. What do you call it? Primary spermatocyte. So, this is that particular cell, okay, which is formed. And what happens to the A cell? A cell will again divide itself and make more and more spermatogonia. So, the A cell will make more spermatogonia, the B cell will undergo differentiation and form the primary spermatocyte. That is what I mean by some of the spermatogonia called primary spermatocyte periodically undergo meiosis. So, what happens to this primary spermatocyte? It will now start undergoing meiosis, and meiosis has how many stages? 2 stages, right? So, in first stage, it will divide into 2 cells. Now, what are these 2 cells known as? So, primary spermatocyte, when it divides into 2 cells, what do you call these 2 cells as? You call these 2 cells as secondary spermatocytes. So, primary spermatocyte and then secondary spermatocyte. Now, you tell me, if primary spermatocyte had 46 number of chromosome and they are undergoing meiosis 1, first stage of meiosis, will the daughter cells will be having 46 number of chromosome or will it get reduced? Isn't it the reductional division? Yes. So, it will be 23 here and 23 over here as well. Yeah. So, at the end of meiosis 1, what do you get? You get secondary spermatocytes. You get secondary spermatocytes. And what is the number of chromosome over here? You had 23 number of chromosome. So, if this was diploid, if primary spermatocyte was also diploid since it has 2n number of chromosome, what is the ploidy of secondary spermatocyte? For the first time, the ploidy is different. It becomes what? This thing becomes what? Half. Okay, the half of diploid, it is known as haploid. What do you call it? Haploid. So, if it was 2n over there, now it is just what? N over here. If this is 2n, haploid means half the number of chromosome. It will be just n. So, 46 becomes 23. Who has haploid nature? Secondary spermatocyte. Okay. What about primary spermatocyte? 46, because it did not undergo any division. It was just getting differentiated. Fine. Now, we are stuck in this particular stage, that is meiosis 1. At the end, what did we get? Secondary spermatocyte. Let's see. So, the primary spermatocyte completes first meiotic division, that is reductional division, leading to 2 equal haploid cells known as what? Secondary spermatocytes. Okay. Now, that is done. What happens to the secondary spermatocyte? Still, it has to do meiosis 2. So, let's see it. Which have only 23 chromosomes each. So, obviously, secondary spermatocyte, as I said, it's haploid, it has 23 chromosomes each. The secondary spermatocyte undergo the second meiotic division to produce 4 equal haploid spermatids. So, basically, what happens? This cell will divide into 2, this cell will divide into 2. At the end, how many cells are there? 1, 2, 3, 4. 4 cells are there. What do you call these cells? Since it is not clear there, I will write it for you. It is known as spermatids. What do you call it? Spermatids. Okay, it is known as spermatids. You have your NCERT. Please go read. Please give a read. Okay, it is known as what? Spermatids. Now, you can remember spermatids as kids. Okay, they are not mature enough. Okay, so the spermatids are transformed into spermatozoa or sperms. So spermatids you can remember as round cells, but sperm, are they round? No, it is elongated structure such that it can easily move. So this transformation of the spermatids to sperms or spermatozoa, that is the last stage. Over here we have something which is known as spermatozoa. I'll write it again, spermatozoa. So there is a transformation phase from spermatids to spermatozoa. What do you call this process as? You call this process as spermiogenesis. Okay, spermatogenesis means formation of sperms. Fine. Now only this process where the kids are become mature. Okay, you call this spermiogenesis. Spermio, this formation of sperm, spermiogenesis. You can remember like that. Okay, this formation of sperm as in formation of sperm from the spermatids. Okay, now that's not all. After spermiogenesis, sperm heads become embedded in the Sertoli cells. So Sertoli cells were there for providing nourishment, right? So now the sperms are going to embed its head in the Sertoli cells and are finally released from the seminiferous tubules by the process called spermiation. So there are a lot of processes being present inside there, but as of now we don't need to dig deep into that. But you just have to remember, after a transformation, okay, the Sertoli cells are there for providing nourishment and everything, and later finally the sperms will be moving out of the seminiferous tubules. Okay, and this process is known as what? Spermiation. This process is spermiation. So children, what did we start with? We started with spermatogonia. Okay, how many number of chromosomes there? 46. It will undergo mitosis to increase the number. Now we have A type of cell, we have B type of cell. The B type of cell will undergo differentiation. Okay, it underwent differentiation, and what did it give? It gave primary spermatocyte. Is it any type of cell division? No, it just changed its structure somehow and it has now differentiated. This particular cell is known as primary spermatocyte. This primary spermatocyte, what is the ploidy of it? It is diploid. It has 46 number of chromosomes. Now it will undergo meiosis. It will give you two type of cells. What are these cells known as? Secondary spermatocyte. What is the ploidy? Now meiosis 1 has taken place. Obviously reduction has to take place. So now the ploidy becomes half. It is haploid cell. So secondary spermatocyte will have 23 number of chromosomes, right? Now secondary spermatocyte will undergo meiosis 2. Okay, which is almost like mitosis. Here there is no halving of chromosomes and all. So you will get at the end 4 cells. Okay, 2 from each. So in this way you get 4 cells. What do you call these 4 cells as? Spermatids. What is the ploidy of spermatids? Is this haploid, diploid? It is haploid because you had haploid secondary spermatocytes, right? Now the spermatids are round shaped cells. You need to make it elongated and everything. What do you need to do? The cells need to undergo a process known as spermiogenesis, where they will transform into spermatozoa or sperm. Now the spermatozoa goes and embeds into the Sertoli cells and gets nice massage over there, and later moves out of the seminiferous tubule, and this process is known as what? Spermiation. I hope this is pakka clear to you. Yeah. Now that is also done. Let's go ahead. What is the correct sequence of sperm formation? You have to tell me. So first one, spermatogonia should be the first one. So I can clearly cut this option off. Okay. After spermatogonia comes what? You tell me. Is it spermatocyte, spermatozoa? What is it? Spermatozoa, I think it's the last one. Can I cut this off? This option is gone. Spermatocyte is correct. So this option and this option we are having. After spermatocyte, what do we have? Kids are there. So what are those kids? No, this is spermatozoa. This is wrong. Spermatids, the kids are there, which will be later transforming into spermatozoa. So this is the correct option. It came in NEET 2013. I hope this is also clear to you. Okay, now, this is also very important. Let's see. Spermatogenesis starts at the age of puberty. Now you might be thinking, why did all of this start at the first? That should be the first question. Basically children, at this point of time in our brain there is some part of it which is known as hypothalamus. What do you call it? You call this particular part as what? Hypothalamus. Now if I can, okay, oh, come on, Just a second, if it is possible by any chance to zoom this. Okay. You see, there is hypothalamus. What is this? Hypothalamus is a very important part of our brain. Just a second. I'm so sorry. Yeah. Hypothalamus is going to release something that is known as gonadotropin releasing hormone. What is it? Gonadotropin releasing hormone. With love I call it GnRH. Okay. Now, hypothalamus is actually best friends with pituitary gland. Pituitary gland is also present in the brain only. Okay. So, pituitary gland, there are two types, two parts of it, anterior and posterior. Okay. So, basically hypothalamus is best friends with anterior pituitary gland. So, hypothalamus releases gonadotropin releasing hormone. This goes and acts on the anterior pituitary gland. Now, the anterior pituitary gland releases two hormones. One is luteinizing hormone and the other one is follicle stimulating hormone. Okay. One with love I call it LH and other with love I call it FSH. Luteinizing hormone and follicle stimulating hormone. Don't worry, we'll write it all. Just try to understand as of now. Okay. Now, let's forget this FSH as of now. Let's focus on LH. LH will go and act on the Leydig cells. Where are the Leydig cells? Outside the seminiferous tubules. Now, what is the use of Leydig cells? To produce what? Androgens. Remember? So, basically this LH will go and act on the Leydig cell. Now, in the Leydig cell there will be certain changes such that the cholesterol which is present inside will be helping in the formation of the male sex hormone or the androgen, which is known as testosterone. So, what happened? GnRH came from hypothalamus, gave a response to anterior pituitary gland, anterior pituitary released LH and FSH, LH went and acted in the Leydig cell, brought some changes such that cholesterol get converted to testosterone. Now what happens? The male hormone which is released by Leydig cell will now not stop but get inside the seminiferous tubule. It was outside, now it will slowly get inside the seminiferous tubule. Now, why do we need it here? Because for the whole process to happen, children, you need testosterone. You definitely need testosterone. But there is a catch. Testosterone will move around. But we need someone who can hold it in one place so that this particular process can take place easily. Now, who is that particular thing? It is ABP. What is ABP? Androgen binding protein. So, testosterone is an androgen. Okay. So, one protein that binds the androgen and keeps it at one place. Androgen binding protein is that. Okay. Now, where did this come from, ma'am? So, basically now comes the role of FSH or follicle stimulating hormone. Follicle stimulating hormone goes and says, hey Sertoli cell, this is Sertoli cell, okay? This one cell that you see over here, it is Sertoli cell. It says, hey Sertoli cell, release what? ABP, that is androgen binding protein. Now, the androgen binding protein are being released. It's being released so that testosterone stays at one place and the whole process happens very easily. Now, you just imagine that this process never took place. At puberty, none of these processes will be taking place as well. Okay. So, this is the thing which was there over here. Now, let's read and try to understand this. Okay. Let's see. I'm going to use a highlighter this time. Spermatogenesis starts at the age of puberty. Why? Due to significant increase in the secretion of gonadotropin releasing hormone, which is known as GnRH. Okay. If you recall, it is a hypothalamic hormone. That is, hypothalamus releases it, which is why it is known as hypothalamic hormone. The increased levels of GnRH then acts on the anterior pituitary gland and stimulates two gonadotropins, two hormones, okay? Two gonadotropins, that means the things that can act on the gonads, that is testes and ovary, okay? What are these gonadotropin hormones? Luteinizing hormones. The spelling of it, if you are not able to see, I'll write it down for you. The spelling is luteinizing hormone, okay? LH and follicle stimulating hormone. Follicle stimulating hormone. Fine. Now, LH acts at the Leydig cells and stimulates the synthesis and secretion of androgens. As I said, LH is going to act on Leydig cell and produce what? Androgens. What is this androgen? Testosterone. Now, we go ahead. What do we have? Okay. And androgens in turn stimulate the process of spermatogenesis. This particular testosterone is important for this particular process to happen. FSH now acts on the Sertoli cells. FSH is acting on Sertoli cells and stimulates secretion of some factors. What are these some factors? It is what? ABP or androgen binding protein. I will write that also. It is what? What are these some factors? Androgen binding protein. ABP. Okay, which helps in the process of spermiogenesis. What is spermiogenesis? The transformation of spermatids to spermatozoa. I hope it is clear. If this is not, obviously if the ABP is not released, the testosterone won't be here, and if it is not here, this particular process will be majorly hampered. Now, you know what, basically there are certain hormones which are being released. Okay, the testosterone itself goes and says the Sertoli cells, please stop this thing that they can do. So basically there are certain hormones like inhibin which is released. Inhibin goes and acts on hypothalamus and says please stop releasing it, so that none of this process could take place as well. So there is a negative feedback as well. This process is known as negative feedback. When something is too much, it goes, our body will be releasing something else which will go and tell that stop, it's time to stop. Yeah, I hope this is also very much clear to you. Yeah, now we go ahead. Sertoli cells are regulated by pituitary hormones known as, which is the pituitary hormone that works on the Sertoli cells? You tell me. LH, I think LH, luteinizing hormone, works on lady cells. LL, right? FSH, I think it is true, right? Follicle stimulating hormone. It will go and act on Sertoli cells and produce what? ABP, androgen binding protein. It came in the year 2006. So that's all. And now finally, and now comes the most important thing, that is what? The structure of the sperm. So if I just tell about the structure of the sperm, obviously you won't be able to understand. We have to draw it. That is why I have this particular space over here. But before that, let's read at least a point. That is, it is a microscopic structure, which means you cannot see a sperm with the help of your naked eyes. Yeah, you need a microscope. Now, what all parts are there? There is head region, there is neck region, there is middle piece and tail. And what all do these regions have in particular? If you are asked to draw the diagram of sperm, you should be able to draw, right? So, for that, I will help you. First thing we need to draw is what? The head region. Let's see, concentrate on the head region. So, over here, in the head region, you have something like this. What is this something? It is known as the nucleus. Now children, the nucleus is very much condensed. The nucleus is what? Very much condensed, because obviously the sperm cannot be big. The structure, yeah, it has to travel smoothly, easily. So it has to be small, elongated structure. So we have a very condensed nucleus right at the top, and above it we have another very important structure. Okay, above it we have another very important structure. This particular structure that you see, it has a name. You call it acrosome. What do you call it? You call it acrosome. Fine. Now, acrosome is a structure which is filled with enzymes. Why do we need enzymes here? Basically, the sperm will be going and fertilizing or fusing with the egg, right? Now, when this fusion happens, obviously the sperm needs to get inside the egg. For that, it has to lyse or break down the membrane of the egg or the ovum. So, for that, what do we need? Basically, they need a set of enzymes. And together, these set of enzymes, they have a name. What do you call them? You call them sperm lysins. Okay, lysins as in they are responsible for lysing, for breaking down things. So, sperm lysins, it's a very important thing. Okay, you need to remember. Now, that is how the sperm is basically able to go and fuse with the egg and get inside the egg and fuse, have the fertilization take place. Okay, now that is about acrosome and nucleus. We go ahead. There is a very small chunnu structure over here. This is a centriole. This is what? A centriole. You might have heard of centriole before when you study cell chapter. centriole is responsible for formation of spindle fibers. Now just imagine like Spider-Man, how we have, right, certain fibers are there which goes and gets stuck. Similarly, there will be centriole which will be releasing out some spindle fibers, and these spindle fibers goes and attaches to the chromosome and pulls them apart during cell division. So you need this particular centriole for that. Once the fusion takes place with the ovum, there will be obviously a cell division taking place. At that point of time, you need spindle fiber formation. So for that, we have one centriole over here. What do you mean by one centriole? Are there two centrioles? Yes, there are two centrioles. So this one, which is near to the nucleus and everything, you call it proximal because it is in proximity. Okay. So this centriole, what do I call it? I call it proximal centriole. Okay. Proximal centriole. Now comes the next thing. What is the next thing? Let's see. So over here, for making it very easy, I'm just making 2 bars. Okay. And then I'm going to draw something like this. Some bands like this. Now what are these bands? What do these bands have? Children, these bands, they basically have lots and lots of mitochondria. And why does the sperm need mitochondria? Because you know the sperm also has a tail region, and for the movement of the tail region, it needs energy. And who provides energy? Where is the energy molecule being formed? Obviously in the mitochondria. So don't they need mitochondria? Obviously. They definitely will be needing mitochondria. So in these bands, they have a specific name, lubricant band. So basically, over in this bands, what do you have? You have mitochondria. Can I write this? I'm not making everything up. I'll just write mitochondria for you to remember at least certain things. So there are lots and lots of mitochondria, and I'll try to join this and this structure. Okay, like this. Fine. Now we go ahead. Now I told you there are 2 centrioles. So one centriole was there, the other centriole is right over here. And what is the role of this particular centriole? This particular centriole will be responsible for the formation of the tail flagella, which is made up of microtubules. So basically, for formation of this particular tail region, you need the next centriole, which is known as what? This is proximal, then this will be what? It is in distance, so it is known as distal centriole. Can I call it distal centriole? Fine, I hope it works. Now as you know, this is the tail region. Now very important thing, if you think that your sperm looks like this, almost like a rocket, it's not like that. Okay, there are specifically yeah, almost it looks like a rocket, but there are certain changes as well. So your whole sperm, it cannot be left like that. Obviously it's a cell, right? Obviously it is a cell, so it has to be surrounded by what? The plasma membrane. So there will be a plasma membrane which surrounds, why does this come? Yeah, which surrounds the whole sperm and including the tail, okay? So over here you have a structure like this, which goes like this, and you have a tail region. So this is just a tentative figure I'm talking about, okay? Please do not judge my artistic skills with this thing. Obviously when you stand and draw, it becomes messed up a bit, so, or maybe a bit more. So this particular structure that you see over here, which covers the entire sperm, what do you call this? You call this the plasma membrane. It is what? The plasma membrane. Will you remember it? That this particular layer covers the whole sperm. Now for addition, there are few more things. So this is a normal thing. First we will read out, then I'll tell you something that is extra. Okay, so yeah, now I can divide the whole sperm into different categories. Let's see. This particular structure that you see over here, what do I call it? It is the head region. Okay, this thing that is including nucleus and acrosome together, it forms the head region. Now this thing that you see over here, it becomes what? The neck region. This is the neck region over here. Now if you talk about this particular region, what is this particular region known as, which has so much amount of mitochondria? You call this as middle piece. Okay, you call this as what? Middle piece. So this is 1, this is 2, this is 3, and at the end, towards the end, what do we have? What is this thing? This is your fourth part, which is the tail. Which is what? The tail. I hope you got it. Now let's read it. So the plasma membrane envelops the whole body of the sperm, which you just saw. The sperm head contains an elongated haploid nucleus. Okay, we studied about haploid. Haploid as in how many number of chromosome? Half the number of chromosome. So what is the normal number of chromosome? 46. Half of it, 23. So obviously the sperm will be carrying almost 23 number of chromosome, and the egg also will be carrying what? Not almost, pakka 23 number of chromosome, and the egg will also be carrying 23 number of chromosome. Together when they fuse, 46 number of chromosomes are balanced, right? So this is what? Haploid nucleus. It cannot be diploid, it is half of it, so it is haploid nucleus. Okay, the anterior portion of which, that is about the nucleus we are talking about. The anterior portion of the nucleus is covered by a cap-like structure. What do you call this cap-like structure as? Acrosome. Now what is there inside the acrosome? Enzymes. What enzymes? Sperm lysins. Altogether you call them sperm lysins, right? The acrosome is filled with enzymes that help in the fertilization of the ovum. If acrosome is not there, the enzymes are not present, obviously will fertilization take place? No. The middle piece possesses numerous mitochondria, as I already told you, which produce energy for the movement of the tail region that facilitate sperm motility, essential for fertilization. Now, if the sperm is not motile, can it reach towards the egg and fertilize it? No. So, sperm motility is very, very, very important. Now, the human male ejaculates about 200 to 300 million sperms during one ejaculation. Will you remember it? Human male basically ejaculate 200 to 300 million sperms. You have to remember this term. Now, out of which at least 60%, I am saying out of this thing, at least 60% should be having normal shape and size. Because if it does not have normal shape and size, don't you think that it will affect the motility? Obviously it will affect the motility. So the shape and size matters. And now if you go ahead, 40% of this 60%, okay, you already chose 60% among them which has the normal shape and size. And now out of that, 40% should be showing vigorous motility, should be moving really fast. If it moves really fast, then only it can reach the ovum. So in the female body, what happens? Thinking that it's a foreign particle. Obviously, if something foreign comes inside, even our eyes, we tend to blink and our eyes start swelling up, and there will be so much amount of waterfall being flowing around, right? What is this mechanism? Basically, it is a counterattack. Our body does not want anything foreign to enter, right? So similarly, inside the female reproductive tract also, this sperm is a foreign particle. So obviously, the female reproductive tract will try to kill a lot of sperms. So after that, if you want to move ahead, go ahead and win over there, go and fuse with the ovum, you have to be motile, very much motile. I hope you got this thing. Now, as I promised, there are few certain extra things. Let me teach you that as well. So the 2/3 of the head region, okay, the 2/3 of the head region is basically covered by a thing which is known as, okay, is it visible? I shall write it in black. It is known as, it is a layer, okay, it is a layer and it is known as Galea capitis, okay, just 2/3 of the head region. Now below this if you go, I'll make in purple, from here to here you will have another layer, okay, from here to here, that is the middle piece, the end of the middle piece, you have another layer which you call as manchete, manchete, okay. If you want to remember it, you can remember it, it is important, okay. So you have something that is layer that covers up 2/3 of your sperm head, which is the Galea capitis, and below that what do we have? Manchete. Together if you see the structure, The topmost region is acrosome. Acrosome has sperm lysins which will be used for lysing the membrane of the ovum and which helps in fertilization to happen. Now one more very important thing regarding acrosome. Acrosome basically is derived from something which is known as what? Golgi apparatus. So this is also a very important concept. Acrosome is derived from what, children? It is derived from Golgi apparatus. So all the sperm lysins which is in there, obviously it is, everything will be coming from the Golgi apparatus, and the RER makes the enzymes and all these things. Okay. Now below the acrosome we have the nucleus. What type of nucleus? Haploid nucleus. Below it we have proximal centriole which will be responsible for spindle fiber formation. Below it you have mitochondria, bands of mitochondria, and then below it you will have a flagellar tail, okay, which is made up of microtubules. So over here covering all the whole body, what do we have? We have the plasma membrane. I hope every piece of information over here is covered. Apart from that, we learned that there is an extra layer covering 2/3 of the sperm head which is known as Galea capitis. Towards the lower end, what do we have? We have another layer which is known as manchet. Yeah, 200 to 300 million sperms are being ejaculated during one ejaculation. Yeah, out of which 60% should be having normal shape and size, and 40% of them should be having vigorous motility. Clear? Now let's see the questions. Okay. Which one of the following statements about human sperm is correct? Let's see. So acrosome has a conical pointed structure used for piercing and penetrating the egg, resulting in fertilization. I don't think it's so pointed that it can go in and cleave the egg. It's not like that. So this particular statement, even if you read it, okay, you will come to know that it is entirely wrong. So it has nothing to do with the pointed structure and anything like that. So an acrosome has a conical pointed structure, does not make any sense, right? Now, the sperm lysins in the acrosome dissolve the egg envelope, facilitating fertilization. Let's keep this on hold. See, acrosome serves as a sensory structure leading the sperm towards the ovum. No, it is not a sensory structure. Now, the acrosome serves no particular function. That is obviously very much wrong. So what is the correct answer that we have? That is, it has the sperm lysins which is responsible for dissolving what? The egg envelope, the outermost layer, which will facilitate in fertilization. So this is the correct answer and it came in 2010. Fine. Now we go ahead for the next question. What do we have? Let's see. Which one of the following statement is false in respect of viability of mammalian sperm? What do you mean by viability? Viability means the ability of the sperm to fertilize the egg. It has not, by reading this question, sometimes people think that it is about how long the sperm could live inside the female reproductive tract. It's not like that. Viability means the ability of the sperm to fertilize the egg. Now, first statement is sperm is viable for only up to 24 hours. Okay, let's just read the options. B. Survival of sperm depends on the pH of the medium and is more active in alkaline medium. Is it correct? Obviously, that is why what do we have? Along with sperm, we have the seminal plasma, which is a secretion of all the glands, right? So, why do we need that? Because the sperm needs an alkaline pH to sustain. In the female genital tract, it is all acidic. Can the sperm sustain there? No. So, surrounding it, protecting it, what do they have? They have the seminal plasma, along which makes the pH around 7.3 to 7.5, which is alkaline. Now that is true. What do we need to find out? The false statement. Next thing, viability of sperm is determined by its motility. So obviously the ability of the sperm to fertilize the egg is determined by motility. If it is not motile, can it go and fertilize? No. So again, this is a correct statement. Now D, sperms must be concentrated in a thick suspension. Obviously it has to be protected, it has to be there in a thick suspension, so this is also correct. Now even if you do not know about the viability, the time duration, you will still understand that these 3 points are correct, but this is wrong. Now how this is wrong? Because the viability of sperm ranges from 48 to 72 hours. It depends on different articles, there are different answers, but yes, it is said it goes up to 72 hours mostly. Now, if you are asking about how long the sperm can survive inside the female genital tract, that is different. It can survive up to for 5 days also. Okay, so viability as in how long it is able to fertilize the egg. That is something different. I hope it's clear. So the correct answer will be what, children? It will be this. Now, with this we have officially completed all the topics. And I want to give you a very brief idea. As I said, since we are talking about NEET, we need to learn certain extra things, okay? So things that I missed over there, I can just explain it to you over here. So over here, just think that this is the abdominopelvic cavity and here is the scrotum, okay, which has what? Which has the testis. Now children, there is an area like this, okay, through which the testis moves downwards. So what happens when babies are there, okay, when boys are babies, okay, at that point of time, the testis is located inside. Now when I'm not saying babies as in when the particular human being is inside the womb, is inside the uterus, at this point of time when it is in the fetal stage, the testis is basically inside the body. And now towards the end, when parturition happens, when the baby finally comes out, what happens? This testis naturally comes down into the scrotal sac, okay. Now what is this space known as through which it comes down? This particular space that you see, it is known as, please remember it, it is known as inguinal canal. It is known as inguinal canal. Now mostly naturally it comes down. For some children, they have to undergo surgery, okay. Sometimes it is not possible even by surgeries. At this point of time, it becomes malignant, okay. It is a problem, then you have to find a solution for that. So yes, so this is inguinal canal. I hope you remember this. Now in males, you know that during the temperature regulation happens, sometimes the testis naturally moves up. It's all through the inguinal canal. Please remember that. Now we studied regarding testis that testis has dense coverings. What are those coverings? Let's see the name one by one. Over here, if this is a testis, can I remove this? I think I can remove this, yeah. Over here, if this is a testis, then there is a layer like this. This is the outermost layer. What do you call this outermost layer as? This is tunica vaginalis. This is the outermost layer. So as you see, there is inner side, there is outer side. The inner side is known as the visceral layer. It is inside. And the outer one over here, you call it parietal layer. Please don't feel dizzy after studying this. It's very easy. I'm telling you, trust me. Now, why there is a fold? Because you need space in between, and in this space there will be fluid filled. This particular space is basically filled with fluid so that the friction won't happen, okay, so that the friction does not happen, which is why there is a fluid in there. And inside this you have the testicular lobules and the seminiferous tubules. So the first layer will be what? The tunica vaginalis. Now we move inside. You know that there are testicular lobules, right? 250. How many of them? 250 of them. Now in between this there will be fibrous connective tissue layer, okay. There will be a connective tissues layer which you call as, what do you call it? This is the second one, and you call this particular layer as tunica albugenia, okay, which comes over here and forms a very thickened structure which is known as mediastinum. I'm not going deep into that, but yes, this particular fibrous connective tissue helps in partitioning the whole testis into different compartments and helps in the formation of the testicular lobules, or else this particular partition would not have taken place very efficiently. So this is the second layer. Now the innermost layer which is surrounding the each testicular lobules, it is known as, any guesses? Do you have any guesses? If you have any guesses, you can definitely tell me. This red colored thing, it is known as tunica. Since it is a vascular layer, it helps in the, in bringing up the blood vessels and everything. It is known as tunica vasculosa. So children, how many layers do the testes have? The outermost layer is tunica vaginalis, which is folded and filled with fluid so that the friction won't take place, okay. Next thing, the connective tissue layer, I'll write it here as well. It is the fibrous connective tissue layer, okay, which is known as albugenia. Now The innermost layer, which is vascular in nature and is surrounding all the testicular lobules, it is known as tunica vasculosa. So these three things, I hope and I wish you just remember it. You can just write it down. If you want, you can take a screenshot of this as well. Okay. The inner side of vaginalis is known as visceral layer. The outer side is known as parietal layer. So outside this, what do you have? You have the scrotal sac. Okay, which is where your basically testis resides. That's all. So that was an extra piece of information that I wanted to tell you. Now we go ahead. We have a few more PYQs. 2 I'll solve, 1 I'll give you to solve. So select the correct sequence for transport of sperm cells in the male reproductive system. Can you select? Since we have already studied about the accessory ducts, we should be able to do it. So let's see. The first one, okay, I think it should be what? Seminiferous tubules. Obviously, it has to start with seminiferous tubules. So let's cancel these one out. What do we have? Over here we have seminiferous tubules, and over here also we have seminiferous tubules. Just a second, guys. Just give me a second over here. So I thought I have already done this question, which is why I went and checked. Okay. So for transport of sperm, first one should be seminiferous tubules. Obviously you cannot put testis there. Even if you are putting testis, it is not a duct, right? So let's start with seminiferous tubules. After seminiferous tubules, what was the thing? You had tubuli recti, remember? After tubuli recti, rete testis. After rete testis, RVEV, vasa efferentia. After vasa efferentia, epididymis. After epididymis, last V, vas deferens. So basically, come over here, let's see. Seminiferous tubules is there, fine. Rete testis is there, fine. Vasa efferentia is there, good. Epididymis is there, good. Vas deferens is there, perfect. After vas deferens, seminal vesicle is coming, vas deferens is coming, common duct, ejaculatory duct, going into what? Urethra, and at last what do we have? Urethral meatus. So isn't this the correct answer? This is C. It came in NEET 2019, not very old. Okay. Now the next question that we have is, if for some reason the vasa efferentia in the human reproductive system gets blocked, the gametes will not be transported from. So the question is, read the question, please read the question. Until then I will write certain things. Please read the options that you have over here. It is saying vasa efferentia is basically blocked. Then the gametes will not be transported from. The gametes will not be transported from. So what's there before vasa efferentia? We studied RVEV, right? So this is vasa efferentia. We remembered E for exit, right? And vas deferens is over here because D is in distance. So what is before V? We have rete testis. Now what is after VE? What does E stand for? Epididymis. So is there an option like this? Let's see. Testis to epididymis is there, okay. Epididymis to vas deferens is there, it cannot be possible. Ovary to uterus is there, obviously not possible. Vagina to uterus, this is all female reproductive things, okay. So what do we have? The closest option that we have is what? Testis to epididymis. Obviously, whatever is made inside the testis is not transported outside into the epididymis. Okay, so rete testis comes in the testis, right? So this will be the most appropriate option. Okay, this came in 2011. Now going ahead, what do we have? This is the last question that we have, and I am keeping this question for you. If you know the answer, put down the answer in the comment section. Okay, and let's see what happens. If you're not able to do, don't worry, I'll be explaining you in the next session possibly, but you have to try and put down in the comments. We will pick one comment for sure. Yeah, so you can take a snip of it if you want. I hope that this particular video that we gave you, it was of great help to you and you understood the concept as well as the questions that I presented for you guys. And if you have any doubt regarding the concept, please feel very free to comment it down. I will clear your doubts in the best possible ways that I can. Right, and also let me remind you, apart from these 5 questions that we are giving you, there is also NEET Premier League series, that is what test series, where on every Sunday we will be giving you 30 questions related to topics that we covered in a week. So you don't need to worry about that also. You will get a paka revision for all the topics that you have covered all together. Okay. And if you are liking this concept that of what we are doing, please do not forget to subscribe to the channel and comment all your good regards down in the comments. We will be definitely reading it, and do not forget to solve the question as well. Bye bye. Take care.